All right, class. So let's do a quick review. If you look at the picture on the right hand side, top, we are looking into the vestibule of the bony labyrinth, and deep to that we see part of the membranous labyrinth, two structures called utricle and saccule. If you look inside the utricle and saccule, you see a raised membranous structure, and that is called the macula. If you blow up the macula, you do see the hair cells in pink, and in between the hair cells there are supporting cells in purple. Now the hair cells, if you look at the hair cells, each hair cell has a long structure, and this is called kinocilium. And there are many small bristle-like structures, and the bristle-like structures are called stereocilia. Singular cilium, plural cilia. So each hair cell has kinocilium, each hair cell has one kinocilium and multiple stereocilia. And if you see these bristle-like kinocilium and stereocilia, they're embedded within a gel-like material. Do you see this thingy or this layer? This is called otolithic membrane. It's a jello-like material that is present superficial or on top of the hair cells. All right. So what's the function of this utricle and saccule? They help us understand, or they help us to sense acceleration and deceleration. So let's take a look at this picture. On the left hand side, we see a woman and her head is in upright position. Now, when our head is in upright position, the bottom picture shows how the hair cells and the otolithic membrane is oriented. Right now, you see that the hair cells and the supporting cells are there, and the kinocilium and the stereocilia, they are erect, and the otolithic membrane is covering these cells. I also want you to notice that the axons coming out of the hair cells, do you see the yellow axons coming out of the hair cells? They come together and they form vestibular branch of the vestibulocochlear nerve, which is the cranial nerve number 8. All right. However, let's say when the head is tilted downward, what happens? When the head is tilted downward, as you see, due to gravity or due to the force of gravitation, the otolithic membrane shifts inferiorly. And as the otolithic membrane shifts, look at the stereocilia and the kinocilia on the hair cells. They bend. Are they bending? Yes. As the stereocilia and the kinocilia bends, that sends out action potential via the axons of the vestibular branch of cranial nerve 8, which then goes to the temporal lobe of the brain for further processing. So let's review. When the head is in upright position, we have the hair cells and the supporting cells in position, and there is otolithic membrane on top. The hair cells are not bending at this point. However, when we tilt our head downwards due to the force of gravity, the hair cells bend. As the hair cells bend, there is more neurotransmitter, or we can say that the axons of the hair cells carry the action potential to the central nervous system, specifically to the temporal lobe of the brain for further processing. And that's how we know that that our head is tilting downward. All right. So we saw that the utricle and saccule within the vestibule helps in detecting acceleration and deceleration. We are moving on to the semicircular canals, and within the semicircular canals we have the semicircular ducts which help us to detect rotational movement. Say no. When you say no, your head is rotating, and that is sensed by the semicircular canals. On the picture on the right top, you do see the semicircular canals in 10. Deep to that, you see the blue tube-like structures, and those are semicircular ducts. Now, at the base of the semicircular duct right here, do you see the swollen region? These swollen regions are called ampulla. What are they called? Ampulla. And I want you to focus within each ampulla. Let's say this one. If you focus inside the ampulla, do you see there is a barrel-like structure? That barrel-like structure is called cristae ampullaris. And right now, this diagram has taken this barrel and zoomed in, and we will look into the cristae ampullaris that lies within the ampulla of the semicircular canal. All right, very similar to what we saw on the macula of utricle and saccule. Now, at the bottom, right here, this part is called cristae ampullaris, and the cristae ampullaris has hair cells in purple. These are the hair cells, and there are pink colored supporting cells. And the hair cells are embedded within a gel-like structure called cupula. So this gel is cupula. And the concept is same. Say no, move your head. So as you rotate your head, the hair cells bend. As the hair cells bend, the action potential travels via, do you see the sensory nerve? The action potential travels via the sensory nerve to the central nervous system, specifically the temporal lobe, for further processing. And as the information gets processed, we understand that there is rotational motion of the head. All right, so I want you to know that within the ampulla, there is a structure called cristae ampullaris. The cristae ampullaris is made up of hair cells, and the gel-like structure that covers the hair cells is called cupula. All right, and this helps us to detect rotational motion. Do you see here? This is upright position. As the head is rotating, the cupula bends, and as the cupula bends, the hair cells bend, and that helps us, that sends more action potential down the nerves, and that helps us detect the rotational motion. All right, looking at the equilibrium pathway. So the hair cells in the utricle and saccule are bent by the otolithic membrane, right? And the hair cells of the semicircular ducts on cristae ampullaris, they are bent by the cupula. These information are carried by the vestibular branch of cranial nerve 8, and these information then goes to the medulla oblongata right here. Do you see the orange nucleus? That's called the vestibular nucleus. So information goes via the vestibular branch of cranial nerve 8 to the medulla oblongata, and from medulla oblongata, that information goes to the cerebellum, to cerebellum, and remember, cerebellum helps us balance. The cerebellum helps us balance. Alright, so the key point that I want you to remember here is that within the vestibule, there is utricle and saccule, right? Within the utricle and saccule, you have the macula. Within macula, there are hair cells, and on top of the hair cells, you have the otolithic membrane. Okay, when you look into the semicircular canals, within the semicircular canal, there are semicircular ducts. At the base of semicircular duct, there is ampulla. Within the ampulla, there is cristae ampullaris, and within the cristae ampullaris, you have the hair cells, and the membrane, the jello-like membrane that covers these hair cells, they are called cupula. Remember, the vestibule and the semicircular canal together helps us to maintain balance or equilibrium. Alright, moving on to hearing now. Okay, so when it comes to hearing, we need to focus on the cochlea. So right here, the tan colored cochlea is circled for you. And within the cochlea, the membranous labyrinth, which is in pink, is called the cochlear duct. Remember, the fluid inside the cochlear duct is endolymph. So this is something that I usually draw out on the whiteboard in class. So I'll try to do the same. So we are looking at the ear. We have external ear. This is external ear made up of, external ear made up of pinna, I cannot change color, made up of pinna, and this part is called external, this part is called external acoustic meatus. So these two parts make up the external ear. Then we have the middle ear. This whole thing right here is middle ear. And the middle ear has the tympanic membrane. We also see the three smallest bones, malleus, incus, stapes. These are the three smallest bones of the body. The middle ear is associated with the pharynx by the eustachian tube. Eustachian tube. The eustachian tube actually helps equalize pressure inside and outside the body. Let's say there is too much pressure built up in the middle ear. Some of the air will escape this way to the pharynx, and so that will reduce the pressure in the middle ear. Okay, so it helps. The eustachian tube is important to maintain the pressure. All right, I also want you to know that the external ear, of course, we have air. Here we have air. But when we look into the middle, when we look into the inner ear, which is all the blue parts, I want you to know